Well, good afternoon and welcome. This is Zoom Time Knowledge, a free public education series hosted by the James Madison University Lifelong Learning Institute. Today's presentation is titled Past, Present, and Future Life with Coronavirus. We're joined by Dr. Chris Berndsen, Associate Professor of Biochemistry and Enzymology here at James Madison University. I want to provide a quick introduction to Dr. Berndsen in case you weren't able to join us for our fall program, which was COVID-19 in Perspective. Talking to Chris just before the presentation, we acknowledged that a lot has changed since that fall program. So we're glad that Dr. Berndsen could come back and update us on everything that's new and different since that first presentation. Chris is originally from Virginia and attended Roanoke College for his undergraduate studies. He then did his graduate work in biochemistry at the University of Wisconsin-Madison and postdoctoral work in biophysics and biophysical chemistry at Johns Hopkins University. In 2012, he returned to Virginia and started teaching courses in biochemistry at JMU, and his work in the classroom and laboratory has been supported by the National Science Foundation, the National Institutes of Health, the U.S.-Israel Binational Science Foundation, the Jeffries Memorial Trust for Interdisciplinary Research, and the For Virginia organization. Chris, at this point, I'd like to turn it over to you to lead our presentation. Sue G and I will be in the background. If anyone needs assistance with the Zoom platform, feel free to use the chat window. Your microphones are muted right now, but you can enter questions into the chat window. When we get to the Q&A period following the presentation, we're going to extend microphone access back to you. If you'd like to have questions or comments for Chris, you'll be able to communicate directly. Until then, just know those mics are intentionally muted during the presentation. Chris, welcome and thank you.
Thank you, and thank you to the Institute for affording me the opportunity to come back and join you all and talk a little bit about coronavirus and you know what's going on in the world and what may be going on. I'll talk a little bit about what we know and what's going on right now, and then some of the threats and environmental ideas and concepts that may be driving the future life with coronavirus and the future in general. And I hope to end with, you know, open questions, but also discussion of what might be happening as we move forward in the new normal, whatever the new normal might be for us. So a lot's going on. We've had a lot of news about cases. We've had testing and issues with testing and vaccines. And we've got presentations from various government officials, and we've heard a lot of jargon and words and things like that. And this has been going on for over a year now. And, you know, I'm not afraid to admit that I'm tired of COVID-19. I'm done, really, at this point. I've been giving talks like this to students and others for better part of a year now, and I'm done with it. I love the science, but I want to move on. And so I think that I guess that many of you feel the same way. And I'm showing here, you know, a socially distanced gathering that I'm sure many of you, or some of you may have had, where I've got my daughter and my parents and being masked and they're distanced from each other. And this is what life is like in many cases right now. We're doing the best we can, but it's tough. And I'm tired of it. And I'm looking forward to moving on to something new and not wearing my mask all the time and being able to see my parents and my family and all of those who I haven't been able to see for a very long time. So I'm sure many of you join me in that. When we're thinking about diseases, they've been around for a long, long time, and they've affected us in many ways. This is a great graphic that I found that shows when pandemics were occurring coming forward to the present time here in the front. And then the size of these little spiky balls is the impact it had on human life. And we can see recently we've had a lot that occur that haven't had as great an impact as some of the past diseases, but certainly COVID-19 is a lot larger than some of the ones we've had like Ebola and MERS and SARS. but realize that COVID-19 is just one of many diseases we've been living with in a pandemic state for a number of years now, including Ebola breaking out again in Africa recently. And so we've been living with disease for a long time, and COVID-19 was a little different because we lost control. And it's hard to say we have any control over nature, really, but we lost control of this one, and it had a great impact on our current life. Coronaviruses are really kind of odd to be a pandemic. We haven't had a pandemic caused by a coronavirus that we're aware of since, except for the SARS and MERS and the current one. For the first 70 or so years that we knew about coronaviruses, they were mostly reported in animals, mice and chickens. They were found in humans and mostly caused common cold types of sickness, not usually fatal, usually could be overcome in a week or two. And there wasn't much known about coronaviruses between when they were first coined as coronaviruses in the late 60s up until around 2003. There were only 4,000 papers published. And so there was a real lack of knowledge about the biology and chemistry and the medical impact of coronaviruses. And you compare that with recently, if we look between 2019 and as of last Thursday, there were 74,000 papers published just on COVID-19 and coronaviruses. So there's a huge acceleration of the science trying to make up for the lack of information that we had. And if you look, we've had 3 outbreaks of 3 different coronaviruses in the past 20 years. So they're here to stay, and we're going to be living with them and trying to understand how we can live with them and survive in these times. If we look at why this may have happened, when we look at bats specifically, because bats are thought to be where coronaviruses leaped from bats to humans, and we look at where bat habitats are being encroached upon by humans, and where's a high population density and a high bat population density, it aligns with a lot of the forests that we see in South America, the rainforest area we see in South Asia, into India, and areas of Africa as well. And if we look at where generally it's considered that COVID-19 broke out in the Wuhan province of China, it's right there on the edge of one of those regions. So this wasn't unexpected in the fact that we knew that bats were a reservoir for lots of human diseases. It happened and caught us a bit off guard, and like I said, we lost control of the virus overall. It was a little over a year ago that we had our first COVID death in the US, at least according to the statistics, and we just crossed the 500,000 death threshold a couple of days ago, and that's a huge number of deaths to happen in a short amount of time. And a part of the reason we had so many deaths in that time was because coronaviruses are unusual in that they don't cause everyone to get sick. There's a significant proportion of people who are infected who will test positive, but who never show the outward signs, the fever, the sniffles, and the cough that you would associate with coronaviruses at sort of a minimal level, let alone the more severe symptoms. And so when we think about some of the preventative measures, checking for a fever, or just are you self-diagnosing with the symptoms, they weren't going to catch these asymptomatic carriers necessarily. And we don't have a really good handle on how to detect them beyond those PCR-based tests and the antibody tests that currently exist for those we look for infections. Now, the estimates are that somewhere between 15 and 40% of those who are infected are asymptomatic. And the progression of the virus follows exactly as those who are symptomatic, in that they stay infectious for a number of days post seeing the virus, but they just never develop symptoms, and we never know who has it. And if you think about, there are about 40 participants in this session right now. If 10 to 15 were asymptomatic, that's a really significant proportion, and they are wandering around society, and we don't know who has it. And so this is where the social distancing and the proactive measures like wearing a mask are trying to cut down on the spread and prevent that asymptomatic spread. So rather than just having those who are symptomatic wear masks, having those who are asymptomatic as well, or potentially asymptomatic, is one way that has been trying to limit the spread. But we didn't figure that out until too late in the game to really do a lot to be preventative and get this under control. The virus is largely transmitted in droplets and aerosols. And so even though the virus is relatively tiny, when it gets in these liquid droplets, it's much larger. And this is how masks can help us, is those masks can absorb some of that liquid that you release. It's not necessarily going to be preventative for you getting it from others, but that's where the current proposal of doing the double masks is supposed to help, in that if you have a cloth mask and possibly an N95 mask or some other layer in there, you can try to limit that spread of those droplets, both that you release or potentially release, but also that others release onto you. So masks are important. This is where social distancing comes in. We always had that 6 feet rule very early on. They said 6 feet apart, you'll be okay. They are now finding that these droplets can travel up to 30 feet. And I saw a paper about a week ago that's saying even up to 50 or 100 feet. So, 6 feet was good early on, and it became clear that we need to keep further apart and wear masks. But also good ventilation is very important, and providing air circulation. The classrooms we have here, when we do any in-person classes now, we're opening up the ventilation fans as much as possible to try to circulate the air to get rid of the droplets as much as possible. And this is something that early on didn't know if the virus was airborne or could be dispersed through the air. And as we learned that masks and social distancing helped, and ventilation, and that's contributed to a lot of the decreases in cases that we've seen in the past month or so. What's also weird about COVID-19 is that unlike the early coronaviruses that caused the cold symptoms, COVID-19 also goes down into the lungs, which is more akin to what SARS and the MERS virus do. And those are the early indications of the more deadly forms of the coronaviruses that were out there. What's really unusual about COVID-19 is that it affects both the nasal passage and then also gets down to the lungs. So it's very easy to spread and very easy to have severe symptoms as well. And it's really the worst of both worlds. Cold is easy to spread, and COVID-19 can spread like a cold. MERS and SARS-1 were very fatal. MERS has a death rate of around 40% because it gets deep into your lungs and starts causing real problems. Well, COVID can do that as well. And so this is another issue, and we've seen a lot of diversity in the symptoms that patients have who have COVID from. They get a little bit of cough and some nasal issues to having to be on oxygen and other ways to help them breathe. And the masking thing is actually a really old solution. This is an instrument from the 1920s, right after the influenza pandemic of the early 20th century, where they built wax noses and then put different types of masks on them, and then pushed air that had, or had someone breathe who had the virus through it, and tried to detect what mask would be best. So 100 years ago, they had figured out that masks actually helped prevent the spread of airborne viruses. And when we look at today, the types of experiments that we do, they're actually pretty identical. We just have mannequins that breathe more like a human overall. And this is an apparatus that was developed by a Virginia scientist. This is Lindsay Marr at Virginia Tech, has developed this setup, and she uses it to test different types of cloths that we make masks out of. And one of her recent publications, she had this cool idea to look at, well, what could you do for a homemade mask? And not sewing one, but things around your house that you might use for a mask, like a coffee filter or a vacuum bag. And she found that the coffee filter isn't that great at preventing viruses, but taking apart a vacuum bag, you could make a decent filter out of it, one that was as effective as the high-grade surgical masks or the types of filters that we put into air conditionings and heating units to try to filter out viral particles. And this has been kind of a cool idea that different materials and different materials and different types of masks may be more effective, and that we might be looking for new ways or the new materials that which we could use to make light masks that aren't quite as oppressive as some of the really heavy cloths that we've got out there. And this is still an ongoing area of study. I've been to a couple conferences where the keynote speaker talks about masks for 90 minutes, and it sounds really dry, and it is, because masks do filter out water, but it's really sort of a interesting and ongoing topic that we've been understanding masks for 100 years, and they've been useful, but it wasn't until recently that we had a need to get back to this idea of, oh, hey, covering your face can prevent diseases. And this is something that in Asia, Japan, and China, they often wear masks a lot, and that knocks down some of these diseases. It's not something that's spread to the US, but in the future, we might see mask usage becoming more common, especially if we get into a situation where diseases could potentially be on the verge of going into pandemic or outbreaks again. Now, I want to talk a little bit about the biology and the biochemistry, because that's going to lead us to the vaccines and some of the more modern work on preventing COVID. So COVID-19 is a virus, and a virus is a ball of protein and fat with some DNA or RNA in the case of COVID-19 in the center here. And it floats around in the air and eventually gets into us. And there are these proteins on the surface which give coronaviruses their name, called spike. And these spikes are the key to getting into the cell. So if we think about a virus entering a cell, it has to have some way to get in. And this virus protein, the spike, is the key that fits into the lock on the cell receptor. And a receptor is another protein that's found on all of your cells. And the one you may have heard about is this ACE2 receptor. And so spike matches up, sort of like a puzzle piece matches up with the ACE2 receptor. And that is the first step to getting the virus into your cells. Now, both SARS-1 and COVID-19 use this same mechanism to get in. And so there's some potential that the COVID-19 vaccine may protect against SARS-1. But it's interesting that COVID is much better at unlocking the door. It's much easier to form this interaction between the receptor and the spike protein that allows it to get into cells, which makes it more infectious overall. Now, once the virus binds onto that receptor, it gets pulled into the cell, and then it begins to make more versions of the virus. And so one virus comes in, thousands of viruses leave the cell, and it copies its RNA, and then starts roaming around for new cells to infect and cause more problems. Now, the interesting thing about the receptor for COVID-19 is that the receptor is found all over the body. It's highly found quite prevalently in the nose, which is where part of the symptoms come from, and it may be the reason that you lose your sense of smell, as all those cells are producing virus and they begin to die. But also it's found in the intestines and the lungs and the heart and parts of the brain and the eyes and the kidneys. And one issue with COVID is that the symptoms are somewhat inconsistent. We have these mild cases, we have the severe cases, we have cases where people are having intestinal issues, we're having heart damage, we're seeing changes in the liver and the kidney function. And it might well be because the lock is found on all of these different cells. And so COVID can enter all of these cells and begin to destroy them. And your own genetics and your ability to have COVID unlock the door to those cells is going to regulate which cell types get affected. And we don't understand that yet. This is something that was proposed early on, and still now we have no idea what is going on and why we get these diversity of symptoms. So we've talked about masks as being one way to limit the spread, but they're not 100% effective. There are other ways, and one of those ways is the vaccine. And I'll give you a 2-minute history of vaccines. I could go on for a semester or more just talking about it, but we don't have that time today. Vaccines and using this idea of exposing someone to a less infectious form of the virus or something that looks similar that would induce that immune response has been around for over 300 years. There is evidence dating back to well before 1700 from Africa and what we now call the Middle East and Asia of using pus and parts of cells from people who had smallpox to try to induce a mild case of smallpox in someone to provide immunity. And this is called variolation. And it was brought to the US in the 17 and 1800s and used quite prevalently there. The last part of the 1700s, Jenner had begun to do some studies with cowpox, and he infected a boy named Thomas Phipps and found that when he got infected with cowpox, his boy, he was not able to get infected with smallpox. And then this is when vaccine starts to come out in terms of the terminology. So it's called variolation originally. When we started using other types of viruses to prevent smallpox, we start developing the term vaccine. And then not a lot happens until the 20th century, when we start developing a lot of vaccines using modern scientific techniques like cell culture and fermentation to start developing vaccines against measles and polio and many other diseases. So that today kids are getting a number of shots early in their life to help protect them or hopefully give them lifetime immunity against a lot of these diseases. And in the 1990s, we saw a large explosion of this idea of anti-vax or being against vaccines because of questions about the efficacy and safety of them. And this was something that Andrew Wakefield and his work connecting MMR to autism, which was later debunked, has spurred a lot of, and it persists this day. So there's a significant proportion of many communities that is actually anti-vax. And it's not that anti-vaxxers didn't exist in the earlier times, it's just that more recently we've seen a larger prevalence of them in the population. So the really goal of a vaccine is to make antibodies. And if you look at my background that I've got on my screen, I'm surrounded by antibodies, and I'm hoping that that protects me against COVID. We want to do the same thing. And a vaccine is going to produce the antibodies that are going to protect you against COVID. Now, if you get the virus, your body will produce antibodies against it, and that will give you some protection. Now, what we do in the coronavirus is give a piece of that virus to induce that same response that the virus would give, with the goal being that you don't have to get the virus to get the protection. We just give you something that looks like the virus to give you that protection. And oftentimes, vaccines can provide or stimulate the immune system to a higher level than the actual virus would itself. And so sometimes vaccinations offer better protection than getting the actual infection itself. Now, sometimes vaccines don't work or they're not as effective. Certainly, if the initial dose of the vaccine doesn't induce the immune system to respond strongly enough. And you may have heard some about, whether we should get both shots of the Moderna or the Pfizer vaccine or just get one and give everyone one shot and go back and give the second shot later. The reason that one shot doesn't do enough is that the initial shot doesn't prime the immune system enough to respond. And so that second shot is that booster to try to get up to the 90 or 95% effectiveness that the Moderna and the Pfizer vaccines that are available currently offer. Sometimes protection against viruses doesn't last. This is why we have to get booster shots or re-up your vaccination. It's because sometimes the virus mutates and it loses the piece or loses the ability to recognize that part of the virus because of changes in the structure of the protein that the antibody binds to, and that can cause some issues. We currently don't know the length of COVID-19 immunity. We've only been vaccinating people on a large scale for about 90 days or so. And so I know that Moderna and Pfizer are still in their trials and trying to look at the length of immunity. And we'll be watching the population at large to see how long it lasts. Yeah, this is one of the major questions. How often are we going to have to get a vaccination against coronavirus? And it's just not clear. Now, traditionally, vaccines are produced by a company. They make the part of the virus that is going to be used to prime the immune system, and then they provide it in a dose, and then a medical professional gives you the shot, and then your body makes the antibody. And that's what a lot of the traditional vaccines, HPV is a great example of the traditional vaccine where a part of the virus is given, and that provides you with the immunity. The Moderna and the Pfizer vaccines, however, are very different in some aspects from these traditional vaccines. And this is where some of the questions about them are coming up. The Moderna and Pfizer vaccines, rather than giving a piece of the virus to your immune system and having it recognize it, your body actually produces that protein. And so in these vaccines, you have your lipids and then your mRNA. This mRNA is the instructions for your body to make the spike protein. And then that spike protein is provided outside of the cell to begin making the antibodies. And the questions that you hear are, well, you're making my body make this protein that it's not supposed to make. Is that going to be harmful? And at this point, it doesn't seem like it is. There are minimal indications that your body is not liking to do this process. And so this new way of doing these vaccines has been around in controlled settings, like laboratory settings, for a number of years and used in those settings. But this is the first time these types of vaccines have been used on a large scale to protect against a human disease. And it's really, from a scientific standpoint, it's really exciting. It's a new frontier because we can make these mRNA vaccines faster. In that, a traditional vaccine can sometimes take 3 to 5 years to produce the protein and begin to figure out if it's stable, and then make the formulation, and then go and then provide it in dose form. They can produce these mRNA vaccines very quickly, and then they can change the sequence of the mRNA very quickly as well, so that it can respond to changes in the virus, such as the strains. And so this can happen in weeks rather than years. And it's really quite exciting, the opportunity here. We don't know fully the risks and all of this, but this is some new technology that we really are, the scientists and the medical community broadly are looking and excited about. Now, here are the ingredients, and this is a bunch of chemical names. I show this to my daughter and she's like, I have no idea what this is. I show this to my students and some of them are like, I have no idea what this is. And so, what are these words and what can we say about them? So let's walk through these ingredients, because it's less ominous than it seems when you look at these lists. So we've got the mRNA, that's the first ingredient. That's the instructions for making the virus protein. You're not making the whole virus, just a piece of the virus and just a piece of the protein, if we really want to get into that. This mRNA cannot integrate into your DNA. Doesn't have the ability to change the DNA and who you are. All it is an additional set of instructions to reduce the spike protein. And this RNA gets degraded very quickly in your cells as well. Now, the other these are more chemical sounding names. These look more like the back of the food ingredients or something like that. When you look at these ingredients, these are lipids, which are fats. And what they do is they surround the mRNA. You can't just inject with mRNA and have it get into the cells. You have to provide a vehicle. And so this is the packaging, and it's these lipids, and they protect the RNA as it's going into the cells, but also provide a way for it to merge with the cells and then get into the cells. And then lastly, we've got a couple of pH buffers. These help maintain the environment such that it doesn't get too acidic or too basic. mRNA is a really fragile molecule, and if it falls outside of sort of a sweet spot in terms of acidity, this breaks down and it becomes less effective. And then finally, sucrose. This is table sugar. You may have heard about that we need to keep the vaccine really, really, really cold at minus 80 degrees C or minus 20 C. It needs to be kept frozen. Part of the freezing process, you also want to have some preservatives in there, and that's what the sucrose is doing. And so these ingredients are fairly benign. When I look at them, I see table sugar, acetic acid, sodium acetate. This is, don't tell my chemist colleagues, but this is vinegar. I mean, this is something that you have in you. That's something you eat regularly. Cholesterol, we have cholesterol. This is another type of fat that you can make. This is related to a fat that you can make. Your body has mRNA. So a lot of what's in this vaccine, we already have, or something similar. We have something similar in you already. So the ingredients don't, stand out to me as something that's totally crazy and that we should be concerned about toxicity, at least at this point. And we haven't seen any signs that toxicity is causing a problem. So where are we right now in terms of the Commonwealth of Virginia, and how well are things going? Cases are trending down. We're seeing this downward fall of cases, and this is good. And this is a good sign. And I was really excited to see this happen, and there wasn't a continuation of this upward trend after Christmas. And the Commonwealth is distributing vaccines. Around 30,000 doses are given each day in the Commonwealth, and it depends on what day you're looking at, but on average, we're looking at 30,000 a day. And at 30,000 shots per day, we're looking at a little over a year before every Virginia resident is able to get both doses. So mid-2022, without some sort of change or increase in the rate at this point. At one point, Virginia was definitely headed towards doing 50,000 to probably 75,000 a day, but it's settled in for the past several weeks around 30,000. So right now we're looking at a year plus a little more before every resident will have both doses and be protected. And if you want to look at vaccine distribution, I'm going to just step out and go to this website here for the Washington Post. It actually has a really nice vaccine distribution page. So if you're interested in following this, the Washington Post has this where they tell you who's been fully vaccinated and partially vaccinated. Right now, nationally, it's around 6% is fully vaccinated. Around 13% is partially vaccinated. And partial vaccination does provide some protection. It's not perfect, but it does provide some, but we still want to aim to get everyone fully vaccinated. And then you can go down a little further down and see what state, how each of the states are doing in Virginia. So Virginia is has been covering about 14% of the population, doing pretty well. We could be doing better, but it's doing pretty well. So we'll call that a win right now, and hope that it keeps continuing that trend. So one thing that we're at risk of, though, is vaccine acceptance and getting enough people vaccinated. In Virginia, in a typical year, we think about maybe the influenza vaccine. Only maybe 50 or 60% of Virginians get vaccinated for flu. While that's good enough for flu, for COVID, which is a little more infectious, we need to get in vaccine acceptance to be 80 to 85% to really instill what's called herd immunity, where the population of those who are vaccinated is enough to more or less protect the population that cannot be vaccinated, either because they aren't eligible for the vaccine, and that would be, at this point, children under 16 or so cannot get the vaccine. And so herd immunity is what's going to protect them. It could be those who are immune compromised and aren't able to get the vaccine. So Virginia overall in this survey of who's going to get the vaccine sits in the 65 to 75 range, depending on where you are in the state. It's much higher in Northern Virginia. than it is in, say, Southwest Virginia. But hopefully these numbers will improve. They've certainly improved a lot. It used to be that most of the US was sort of in these lighter colors, but we're seeing more and more acceptance of the vaccine. And I'm optimistic that we'll continue on that trend, because it would be nice to be able to have herd immunity and have this go away. And the students here at JMU are thinking about this problem. I gave an assignment. We had a break day a couple of weeks ago, and I gave them an optional thing to make how the COVID-19 vaccine works posters, and they're going to plaster campus with these things to encourage the students to get vaccinated. And so I've got 80 students in my class right now. Many of them are considering careers in medicine. Some of them are working as CNAs in nursing homes right now. So they are thinking about these problems too, and want to get to the new normal as much as anyone else. And so these are some of the posters they've made. And if you happen to come on campus and see some of them, they were my students, and I'm so proud of them because they did a really good job, and they explained their science really well. And they seem to have a lot of fun doing this too. So we also need to look at the rest of the world. It's great if the US and Virginia are vaccinated, but maybe we want to travel, and traveling will be an issue. And if we look at the timeline, the US aims to have it widely available by early next year, so that more or less we're fully vaccinated. If you look at parts of Africa and Asia, they're looking at 2023 before to be fully vaccinated. So this is a problem that we have local issues, but we're also going to have global issues of travel, and that the coronavirus is still going to be able to be spread quite heavily in these areas that aren't vaccinated necessarily. And so this is a problem that will have to be considered in the future. So cases are moving down, vaccinations are staying consistent, hopefully moving up. What does this mean for the near future? Next month, when we're looking at May or June as school is getting out, what do we need to be doing? Well, we need to keep our masks on for a little while longer, and to keep wearing them. My daughter has wonderful masks that were made by her grandmother, and she loves them a lot, and they really express my daughter's style, which is chaotic all the time. And she's going to school and has been enjoying school quite a lot, in part because she said, "Dad, you're not a very good teacher, at least of kindergarten." So we were struggling for a long while. She didn't want to learn college level biochemistry, and I was having a tough time teaching her basic kindergarten. So she's happy to be back in school and wearing her masks. And we're going to continue that way for at least the immediate future to do this, because even if you have your vaccine, there is the potential that you can spread the virus still. We don't know what that potential is and what the risk is, but we know that it is still a possibility overall. And then we've got this new strains issue, and this is the other reason to wear masks. As viruses acquire mutations, they acquire mutations as they replicate. So when we have this process of getting infected, the virus producing more viruses, it can acquire these mutations. And these can sometimes generate different properties of the virus. It could be more or less infectious, it could be more or less deadly. And COVID-19 has been mutating since the beginning of the pandemic. And I'm going to show a map here of all the different strains that are found in each state as of, I think last week was when I pulled this graphic down. And this has been happening since the beginning. There's a website called NextStrain, and which tracks these mutations. And so this is when we started seeing COVID in 2019, and here's it as it has spread over time. And each of these dots is a new strain starting to diverge from the original strain shown here. And if we watch this happen over time, you are going to see the dots begin to emerge as we go from left to right. And if you watch the right side map, I'll show this 2 times so you can watch each graphic independently. You'll watch how each strain spreads throughout the US. So here we go with the graphic. So we can start seeing divergence in early January and February when more people begin to get sick. And we're seeing there's basically 1 or 2 strains in the US for a long time. And then around May or June, we start seeing some more of these strains appearing, and we start seeing some of the yellow appear. And so every time we see a new color, we are seeing a new strain emerge. And so this is something that has different properties and has acquired a set of mutations. And we keep going. And this is right about the start of Thanksgiving. And keep moving forward. And here's where we are right about today. And there are a lot of strains out there. There are about 10 total that have been observed in the US. 8 of them are currently spreading out there. I'm going to play this one more time. And if you want to watch the right side map over here, it's the US, and you can watch as the virus spreads among the states and watch as each of these little pie charts changes color to indicate a new strain appearing and then taking over. So we get our first cases showing up in Wyoming and California, and they're all generally one strain. And then we start seeing new strains in Virginia and DC. And out west, the green starts showing up more prevalently. And we're starting to get to summertime where we had a real takeoff in the cases, and we start seeing the yellow show up in the south, here in the east, and then it shows up in the Pacific Northwest, and then starts filling in the gaps in between. California only has 3 at this point, whereas some states have 4 or 5. And then we start seeing more strains spreading in each of the states and just going like mad. And eventually we've gone from all blue strains to now all the yellow ones. And we're moving into a completely different set of viral strains, really. And now we're starting to see the red build in as we have the emergence of some new strains that are occurring today. So what does this mean for us? Well, the new strains are thought to be more infectious, and some of the existing antibody treatments and vaccines may not be in fact effective. We are, 3 or 4 months behind the virus in terms of that. However, it might be that, or the vaccines may induce what are called polyclonal antibodies, meaning they're not specific for one target. They can sort of look at lots of different targets and attack different parts, and that makes it harder for the virus to escape. But a lot of the treatments that we have out there may not be as effective, including the one that was given to former President Trump, which they got a lot of attention, that Regeneron antibody treatment that he received. That treatment is very specific to one strain, and now that treatment may not be as effective. And so that is something that we as scientists and medical professionals have now lost from our toolbox to fight back against COVID. So as more strains are developing, what might this mean? It might mean that we need to maintain masking and limiting contact for a little bit longer, just to try to get the cases lower and to really make sure that we can get rid of these infectious strains. It might mean the vaccine has to be redesigned, but that should be easier to do. Or it might mean that we need to get a COVID booster, in about 6 months to 2 years. So there might be a window at which we're getting an update to our booster shot to see what happens. And sort of, this is something that a colleague of mine was spreading, Smokey the Bear was out about preventing forest fires. It's the same way here. We can't get more strains if the virus can't replicate. So by limiting the infection and not getting sick and not being an asymptomatic carrier, we can prevent more COVID variants from coming out and get out of this situation sooner. So what happens next? And this is a part where, I'd love to say that I can predict the future. I can't. But this is some things that we as a community, and both local, at the state, the national, and the global level, should be thinking about as some of the risks. If we look at what UVA is currently predicting as to when the case numbers will be all the way down at the bottom, Mother's Day might be if everything goes well, if we all wear our masks, if we all social distance, if we all don't get that new strain of COVID, Mother's Day, we might start seeing case numbers drop to where we might have no new cases on a day. It might be, 4 or 5 one day, 0 the next, something like that. And then UVA says that by July 4th, we could be seeing basically 0. And that would be fantastic. However, if we get fatigue, and I started out this talk with talking about how I'm tired of COVID, and so many of you probably are too. If we get fatigued, well, then we just keep delaying and longer how long we're going to be here. So I know it's hard to stay strong and wear your masks and be restricted like we are, but the end is so near. We are so near to getting there. And as we get more vaccinations and we keep doing the right things, we're going to be done with this soon. So, while I started out all depressed and all of this, I see a lot of hope in the numbers that are coming out, and I'm hopeful we're getting there soon. But we also need to think about those who remain sick. And I don't mean with COVID, but the long haulers. There is a syndrome that seems that those who have had COVID, about 10% of them experience other symptoms that last long beyond when they're infectious. And this is some sort of semi-permanent and maybe permanent state, we don't even know yet, of general fatigue that seems to be directly linked to COVID. Who knows why at this point? But this is a long-term healthcare situation where 10% of our population could be suffering some sort of symptoms that impair their function in a way that's linked to COVID. And I pointed out that we have a lot of opportunities for humans to interact with animals that might get us sick. And these are called zoonotic viruses, where we have this jump from animal to human. We can get this from a lot of different sources. We saw COVID came from bats. There are other types of viruses that come from domestic animals and farm animals, ones that come from more exotic but sort of natural animals, and then ones that are not typically found here but maybe hunted for various reasons. And there are a lot of diseases associated with those. And as we are expanding and going out into these other areas and interact with animals more, these are the risks of a new pandemic starting. And there was an interesting study looking at animals that had these potential for zoonotic viruses, the ones that can jump to humans, and sort of what classification they fell into, and domestic animals like pigs, and ones that hang out in our houses, mice and rats, and bats, which we have around here, and then some of the other farm animals. They fall on different areas of this cycle. But if you look at rats and hogs, they are quite likely to be where we might find another virus popping out. And not to induce panic, but H5N8 just showed up in Russia. It comes from birds. It's never been found in humans before, and now it's in humans. And so this is something that we need to figure out. And I'm not saying that we're going to figure it out here in this discussion. It'd be fantastic if we could, and be really great. But it's just something we need to think about, how we interact with our environment on a animal scale, and in what we do and how we live can affect so much of the global community. So we've got new strains, we've got new changes. What's going to happen? I don't know. But if we are proactive and are aware of the science and what can happen, and do our best to limit our infections, we're going to beat this, and we definitely are very near. And I thank all of you who are wearing your masks and social distancing, and I hope you continue it, because we are so near, and this has been a team effort, and we're doing a good job, and it's only going to get better. So as we're thinking about this, we might need to get regular coronavirus vaccinations in the future, as I've said. Think about community vigilance and support, remote working, remote schooling. Preventative measures might be something that we always sort of have around. I'd love to say that in 4 months, when this is gone, we can go have a big bonfire of our masks and roast marshmallows over them and have a good old barbecue. But we might need to keep the masks around just in case. And then also support for science and understanding science as a process, and things change. But also what we're doing here. And again, I just said at the top, I appreciate the opportunity to come talk to you all, but having these interactions between the scientist and the community, and you telling me your concerns and me telling you my concerns, and having that exchange is really important. So I thank you all for what you're doing by coming to these presentations and adding your voice to the community. If you're interested in more information, and you're interested in or have access to what are called podcasts, 538.com has a good one called Podcast 19. They do short episodes. They're only maybe 20 minutes, but they do a nice job of breaking down the new science and the data that's coming out, and they try to do it in a way that's very accessible. And they had actually a very funny interview with Dr. Fauci a few months ago, and so I recommend that one if you're looking for something to look at. There also are some interesting books. This one's by David Quammen. He published it a couple of years ago, but he talks about this animal-human interface and where sickness comes from. And it's a very accessible read. Of course, I say that as a scientist with a PhD, but it's an accessible read and is actually fairly clear language about how to understand this and just to think about these potential changes that we're seeing as we move into spaces where we typically haven't found humans before. So with that, I thank you, and I'm happy to take questions. And again, I thank you for all that you're doing and being here, and also your work in the community, wearing masks, social distancing, and taking the time to protect our community as a whole.
Chris, thank you. I do want to point out that there are some questions in the chat window. If you would like to open that as we segue into the question and response period. And after you address those questions, some of those you have covered, but you may need to reiterate. We lost some people who came back. Then I'll open up the microphones if that's okay with you.
So one of the early questions here was, what additional precautions, in addition to masking and social distancing, should we try to take to get back to normal after being fully vaccinated? So one thing that's going to get easier as we move into the spring and the summer is ventilation and staying outdoors and being able to be in areas where there's a lot of airflow. There's that phrase, everything old is new again. This is something that was figured out with the influenza pandemic in the early 20th century, that ventilation was key. And then we appeared to have lost it and picked it back up again. And so in the last several months, there's been a big push to improve ventilation and open your windows or run a fan or something like that to try to just improve the circulation and knock down the concentration, if you will, of the virus that's available. So I would push as ventilation as an additional thing. Beyond that, I think that we're just going to have to ride it out for a few more months to try to get back to normal. So let's see, the next question is here, what vaccines are approved for use in the US and worldwide? So at the US level right now, we have the Moderna vaccine and the Pfizer vaccine, which are offered as emergency use authorizations. They're not actually approved, but they've been given license to distribute in the US under an emergency use. Those studies, the phase 3 clinical trials are still ongoing with those vaccines. So they are nominally approved in that they can be used right now, but the data are still coming in for further approval. The Johnson & Johnson vaccine is up for FDA meeting in about 3 days, I think on the 26th, so Friday. We might have a third vaccine available to us, and that one does not use the mRNA technology. It's a more traditional vaccine, and it appears, if the data are accurate, that it's a one-shot vaccination. So rather than having 2 visits, this will be 1 visit overall. Worldwide, there is a Russian vaccine, the Sputnik vaccine, which is available. And for a while, it was approved before the Moderna and the Pfizer vaccines. There was an industry in Europe and in US of wealthy people flying to Russia to get that vaccine because they could hop in line and get it pretty quickly. So that is one. There's the Sinovac vaccine out there as well, which is made in China. And I think those are all the ones that I'm remembering right now. So there are 2 available in the US. There is another one in Russia and another one in China. They're also available. When COVID began in China, was there a way to detect it moving internationally via aircraft or shipping? That's a really hard question, especially early on. We didn't have a good way to detect it because the tests for the virus were not well designed to particularly go after or those specifics. We really, even now, don't have a good way to track the virus because there are inconsistencies in the random testing that's going on and the tracking. Europe is doing a great job of tracking and keeping track of the strains, more so than the US at this point. I believe the US is making a push to try to do a better job so that we can keep track of the new strains, but also where things are going to try to hopefully localize responses. So maybe, the southeast of the US can start opening up and we sort of watch how the strains are moving so that as we get better, maybe the northeast could open or the Midwest could start opening. So we might be able to stage the reopening of the country, if you will, if we do a better job of tracking and tracing. Is it possible the annual COVID booster and annual flu shot will be given as one shot? That's an interesting idea. I don't know if that would be possible. I'm going to say I don't know is my solid answer. The sort of opinion, as I'm free thinking right now on that question, I think that the challenge would be that two different companies probably make the vaccine. I would love for it to be that way. Wouldn't it be nice and convenient, only get one shot rather than having to get two? But I just don't know at this point. So could anyone try to explain why the percent of deaths per infected people is bigger in developed countries and is not in third and fourth world countries? So one thing that we're finding, and there's some new evidence that I was reading about a couple of weeks ago, correlating weight and BMI to COVID infectivity and the severity of the symptoms. And so it seems that, at least in this early study, that the heavier you are in terms of weight or your body mass index, the more likely you are to start having some more severe symptoms and also be more infectious. So in general, heavier people are going to be found in more developed countries because of the accessibility to healthcare and diet and things like that. So that is one idea for why the deaths might be higher, is because we have this difference in our sociological conditions that are allowing us to be more susceptible because of those conditions. There are some other ideas out there in that it is that we lost control a lot more, and that some of these less developed countries are more prepared for disease outbreaks, especially in Africa, where they are constantly dealing with various new strains of viruses. They tend to be more on top of it with the healthcare to say, okay, don't wear your masks on all of this. And so the other idea is that we as first world countries, if you will, weren't prepared to face a challenge of this nature because we haven't had one like this in a number of years because of the improvements in the quality of life. But again, there are a lot of ideas, and I think that this question right here is going to be one that is going to be debated by scientists and historians and epidemiologists for a number of years. How can humans have a more adaptable immune system when interacting with certain animals and the diseases that can be transmitted from them to us? So different animals have similarities to us, and we are constantly exchanging viruses between different species. And so a lot of the animals, bats specifically, have the same types of proteins that allow coronavirus to replicate, and then they can transmit it. And what you see is the same issue when we have a lot of people around and exchanging diseases, that someone will get sick. It's the same thing when you have a lot of animals around in a compressed space, like farms. Sometimes you have a lot of diseases that can occur. And if the humans come in contact with that, and end up seeing a virus that they haven't necessarily anticipated, or your immune system hasn't necessarily anticipated, you can get sick from that. And that is the potential with this H5N8 outbreak in Russia, is it was found on chicken farms and appears to be going to workers at those farms, and that they've got this interface with an area that is just ripe for lots of viral replication to occur. Yeah, so that's the last question I can see. Are there other questions or things that you want to talk about that I can answer?
Hey, Chris, I have a question for you. This is
Yeah.
Ann Hendrickson, former ISAC professor and geneticist. I have a question for you about something that plagues me only because it personally might affect me, and that is, have there been any genetic studies done that attempt to nail down any specific gene, like gene alleles or single nucleotide polymorphisms that would characterize a person who might undergo cytokine storm or some other really drastic reaction to COVID versus the guy who gets, sniffles and loss of taste and smell? I mean, there has to be some underlying physiological predictive tool that would allow us to isolate the people who would be most adversely affected. And we don't seem to be focusing on that, or I haven't seen anything focusing on that, because that would be a really good way to do sort of triage for vaccines and stuff.
Yes, and that is a great question, and you've hit on 4 or 5 different points that really get at one of the deficiencies that we are currently facing. I agree that we don't have this good sense of the genetics for what is causing infectivity. A couple studies that have been done, and they're being doing a lot out in the UK. One that actually came out of China as well was looking at blood type and trying to connect some of those genes to infectivity of COVID. And that's been a real mixed bag. There have been a couple loci that have been identified, but there hasn't been a lot of follow-up to show that one person is more susceptible than another, and that they can directly link it to these genetic traits. There was a study back in July where they were looking at twins. They found these twins, one who got severe COVID and one who didn't. And they said, that doesn't make any sense. You have a twin, it should be very similar. And they never were able to figure out what was going on. And that, I think, is one of the reasons, is that these twin studies have come out that say that you can have twins that act differently. And it's sort of, pushing back against the idea that there is a genetic link.
Epigenetics.
Yeah, epigenetics is certainly what the next thing to look at. And so I would love to have a solid answer for you and be able to say, go look at this paper, and it's going to be the key to what you're asking about. I think the cytokine storm issues that are out there, and cytokine storms, for those who are, I'm talking science words, cytokine storms are a immune response that is an overreaction to the virus, and has been a problem for a number of, or a significant of the portion of the population. We don't know how to connect that cytokine storm to genetics and the virus fully yet. And I would love to see some more information, but I think a lot of what we're focusing on right now is just trying to get a handle on the virus and how to prevent it. And I would anticipate that for the next 10 years, COVID is going to be a hot topic to try to understand.
Thank you, Chris, and thank you for doing this talk.
Yeah, thank you.
I see that we have Bud has his hand raised. Bud, if you'd like to unmute yourself, you can speak next.
Okay, thank you very much for this presentation. I have kind of a strange question. You hear about the underlying conditions and the people who are especially susceptible. And I'm a kind of a righteous person who thinks, well, I'm very healthy and maybe I won't be affected by this. But the question, I guess, is whether people who don't have underlying conditions and they can still be infected, even though you don't have any symptoms. You could be carrying it and pass it on to other people, right? Is that what you're saying? It doesn't matter if you really are super healthy, you still can be infected easily and can pass it on?
Yeah, and that is what has been so nefarious about this disease, is that we don't know who has it. And it's quite scary from an epidemiological standpoint, and not knowing who is a potential carrier. And this is where looking at the testing and doing a better job to track and trace, and more randomized testing may give us some insight into what asymptomatic carriers have in common. And getting to Anne's question about the genetics, there might be some condition that allows someone to be an asymptomatic carrier and spread it. But you're right, there's a big risk for very healthy individuals spreading this and not being sick themselves.
Okay, thank you.
Yeah, you're welcome.
If we have other questions, comments, feedback for Dr. Berndsen, feel free to unmute your microphone and speak. I don't see any in queue, so you're welcome to just unmute yourself and let us know.
Just got a question in the chat about the twin study, and I wish I could remember the specific details of that. I would have to go back and review it myself. I don't remember what types of twins. I know they were both male, and I believe this was in the Netherlands, but beyond that, I cannot remember more of the details, unfortunately. I'm sorry.
That actually answered it for me.
Okay.
Because if they're both male, they were probably, exact twins, not male and female. Okay, thanks.
You're welcome.
Hello.
Enrique, we have you. You can go ahead with your question.
Okay. Yeah, I have a personal question, and then just to follow up in the other question that I made, I'm the one to ask about the percentage of deaths in the United States against the rest of the world, because I'm still shocked of how people, being packed in India in the same train, and people in Africa, that they don't, the percentage of the people dead are 10 or 20 times smaller than countries like United States and England. That still shocked me, and I know that it's still a debate. And maybe he could explain a little bit. I've been trying to read about the antibodies and the B cells and the T cells of the antibodies, which I couldn't understand too good, and I was expecting to hear something today, but that's one of the explanation. Apparently, some of the third world countries and the fourth world countries, because they don't have access to medicine, they've been producing antibodies themselves because they just get accustomed to face more sickness of different types. And maybe because we're concentrating only in the connections of the coronavirus and not in the nucleus, let's call it like that, of the coronavirus, is because they have developed some kind of defense against it. The other one is personal. What can he advise me? I already had my second shot, and my wife too, 4 days ago. For the past 4 days, we are in bed. We have had a reaction. We really are exhausted and with temperatures, and we cannot even go out of this house. I hope it passes, and we keep thinking, but it's something that we should do to get out of this reaction that we've been having for the second shot.
So congratulations on getting the second shot. That's great. And I know that you're in a bad way right now, but I would encourage you to rest. That's going to be, unfortunately, the best thing. Effectively, your body is fighting back as if the virus were there, and so you feel that reaction because it thinks it's trying to fight a virus, and it's going to be a couple of days, and fluids and rest and do the best you can at self-care, and know that in about 7 or 8 more days, you will have 95% protection against COVID-19. And hopefully that's good. And I know that's no solace for you right now, but hopefully the suffering now is worth it in the long run, not to get sick and the worst things that could happen.
Okay.
when you talk about the other countries, to get to the first part, you're right. There are a lot of inconsistencies, and my mom used to say that the pediatrician told her that I was supposed to eat a chunk of dirt every day or something like that when I was a kid, and that is not a bad idea. Getting exposure to these elements and environment is something that can protect you. And it might be that those countries have low levels of coronaviruses or things that can cross-react with COVID, and it allows them to have some protection that we just don't have here necessarily. And we just don't understand enough, unfortunately, about all of it to really give a better ideas of how we can be more proactive.
And sorry, the last part of that question is, and does anybody have made a study of what is the effects that we're going to have? My wife and I, we've been almost locked for the past 10 months because of several of our problems and our age. But now we have lived in our house locked. And, now we're vaccinated, and, in about 2 months, probably we'll start going out. Are we, influenza, just the normal sickness? Are they going to attack us more because we've been so protected in our house?
That's a great question, and I think that that's another one that is not clear at all, and that is another risk. And I can say that my students have reported that remote learning, they didn't get quite as sick during the fall. They didn't get quite as sick during the spring. I had one student who said she's going to wear a mask a lot more because she's not getting as sick as she would normally during the college semester. So I think that you bring up an interesting problem, that we don't know where that isolation is going to lead to in terms of what the long-term risks could be. Are we missing out on some other disease that was floating around, or a virus or bacteria that was floating around that would have been mildly annoying, but still we should have gotten protection against? It's not clear, but a great topic to investigate in the future.
Chris, I want to echo what you said, that we hope, Enrique, you and your wife get to feeling better soon, and that you have a future ahead that does better protect you against this virus. And Chris, it reminds me where we're at right now. These questions are where we ended in the fall, that we knew some things. There were some unanswerable questions in that moment. We have more information today. I echo something that you probably see in the chat window, just commending you on pulling all this information together. It's incredibly informative, extremely well organized. We get new information, it seems like, every day. And having someone like you who takes the time to not just organize our learning, but to help us interpret all of that information that we've gotten in bits and pieces, to see it all together in a way that makes it very accessible to the public. So thank you for sharing your expertise with our learning community. I want to respect your time and everyone else. Some of them have classes online at 1:30. Sue and I need to host one, but it's always a pleasure to talk with you. Thank you for making yourself available today. And I know that as we learn more, we may be coming back together yet again and saying, here, we're not just 1 year into it. Hopefully the next time we talk, we can say we're 6 months past it. That would be a wonderful occasion to be looking at it in hindsight, instead of looking forward to the future, where we hope it passes swiftly and that we're all well and we can be together again. But until then, everyone take care, and everyone have a great day.
Thank you.
Thank you.